And greetings, everyone, and welcome to this Cell and Gene Live, Driving Down COGS, Automation's Role in Cell Therapy Manufacturing. As Miles said, I'm Erin Harrison, the moderator for this presentation, and I'm thrilled to introduce to you our expert panelists who we will hear from in just a moment. Craig Beasley, Chief Technical Officer at BlueRock Therapeutics, and Narinder Singh, Chief Technical Officer at Arcellx. And of course, thank you to Thermo Fisher Scientific for partnering with us on this event. Now, before we get going, we will be taking questions from our audience, so please be sure to submit them via the Q&A tool. Your questions provide value to everyone here, so I highly recommend that you submit them, and we will get to as many of them as we can. All right, let's get started. So the need for standardization and automation are really critical drivers to innovation in cell therapy manufacturing. An implementation of an automated workflow minimizes open processes, manual touch points, it increases reproducibility, improves traceability. This has the potential to reduce manufacturing failures and lower the cost of goods sold, and therefore facilitating increased patient access, which is what the whole goal here is. So we're going to get into our discussion now, and we want to talk about how automation plugs holes and pushes cell therapy to really new heights within the sector. So Craig, I'd like to start with you. Let's level set the conversation though first. Talk us through the true benefits of automated manufacturing.
I think you hit on many of the key inputs to what helps. I think when you look at CAR T manufacturing specifically, or other types of cell therapy manufacturing, people focus on the individual steps of the manufacturing process, which is great, except really the key to getting scalable is how do you do things concurrently. So automation helps you focus your resources on the critical activities while hopefully automating some of the more standard media prep type activities or kitting manufacturing activities, so that it allows your people to do multiple lots at one time. So that's a key piece. You have to think about that when you're designing the automation. I think the second piece is, at least in our field, is still very new. So we're still collecting data to optimize our processes all the time. So by using automation, you can then get more data out of your processes and correlate that back with the outcome so that you can generate a better process and better automation in the future. So those are the keys to long-term sustainability.
Great. Okay. Narinder, I'd love to hear from you too. What, from your perspective, are the key benefits and the general benefits to automated manufacturing?
I think both you and Craig said it well. Automation has transformed the manufacturing industry in general, right? And even pharmaceutical manufacturing. But when I think about cell and gene therapies, it's a whole different ballgame. We're talking about highly innovative therapies that can potentially cure people with a single dose. That sounds almost close to magic. But these processes are new. This industry is still pretty nascent, and the processes are very complex. And oftentimes we have patient-specific batch manufacturing that is hard to scale and just not cost-effective. At Arcellx, for example, our mission is to advance humanity with our engineered cell therapies. And our goal is to deliver these therapies, not just, make them broadly accessible and deliver them in a cost-effective way. So the challenge for the process people like us is how can we translate this promise to be broadly accessible to thousands and thousands of people, potentially hundreds of thousands of people. And that's where automation can help us, just like it has helped us in other industries.
Good. Okay. And that's actually what I want to talk about next. Now, you mentioned that in terms of process automation for cell therapy, this has the potential to help hundreds of thousands of people if we can get there. So Narinder, what are the key areas, would you say, that need improvement to really scale up the quality and help reduce the costs of therapeutics from a process automation standpoint?
Interestingly, I think in our view, it doesn't start with automation first. The first starts from getting the right construct. So building a research and process development partnership right from the get-go so that we have the right product construct that is manufacturable. So think about manufacturability from the get-go. Once you have the right product construct, then process scientists and engineers can apply the process strategies to improve robustness and efficiencies. And then is when you can overlay automation smartly to improve quality, scalability, and cost effectiveness. So as an example, our research team at Arcellx have really fundamentally re-engineered the CAR-T engineering. And in this case, we have a core CAR construct, which utilizes a small, fully synthetic triple helix-based D domain. And that D domain allows us to have a fundamentally improved transaction efficiency and fundamentally improved CAR expression on the cell surface. For our pipeline, we also created this universal CAR cell product. We call it Arcellx, and you can pair it with different proteins to create different target various antigens. So in this case, we have a manufacturing-friendly CAR construct, and then a universal cell product. So that can be optimized once, and you don't have to keep on reinventing the process every time for different pipeline products. On the process side, I must say that CAR-T manufacturing is hard and challenging, and you hear about in the news all the challenges that they're facing. We've been able to build a process in close collaboration with not just our own quality and tech ops team member, but also our CDMO partners like Oxford and Lonza we've partnered with. We have a process. We've been currently at 100% success rate for patients so far, and which is unheard of in CAR-T manufacturing, like I said before. And the last thing is really having the right partners, right? So from a product partnership standpoint, we have an excellent partnership we just announced last December with Kite, who is a global leader in cell therapy. So in this case, we are learning from them the best manufacturing practices and how can we create the best commercial manufacturing footprint so that we can make our therapy broadly accessible.
Great. Okay. Craig, tell us your thoughts on this. Now, especially given we know Narinder talked about partnership, extremely important. What are the key areas that, from your perspective in general, or from BlueRock's perspective, the need for improvement to scale up the quality and to help reduce the cost?
No, I think Narinder hit a couple key points. Well, first of all, it starts with the process development. Too many companies think about process automation when they've already locked their process and filed the process, and then you have to really question the comparability, et cetera. So it's building yourself enough of a design space that you can automate it. So you need to have media development early on so that you have a wide enough room for error in your media and still have a suitable process so that you can automate those steps. So it's really building and knowing your failure points, because it's so much easier to design something when you know what range you have to design it for. I think the second piece is, especially when you're talking about autologous cell therapy, where it's one patient, one lot means one extra QC test or multiple QC tests. How do you automate the sample retrieval and the QC test? That has a huge benefit in reducing cost and labor and flexibility. So it's not just the process automation, but also the testing automation. Those are really some of the key areas to do it. And I think the third part is, again, it's really trying to optimize through automation the entire process, not individual unit steps. Because when you start optimizing or engineering solutions for one individual unit operation step, it may make it so rigid that you're not able to do multi-processing at a time. So you really have to think, what is your bottleneck? What is your highest cost area? And typically it's not the growth and expansion of the cells, which are easy to automate. It's the time-sensitive steps.
Okay, thanks. And, I want to expand on that a little bit, Craig, if we can. With process automation and process development being, the key component here, talk us through, perhaps for our audience members, what are the best practices that we can offer them, and or the absolute things to avoid when trying to do this correctly?
I think the very first thing that you should do is ask what is critical for your supply chain. in your automation and strategy. I've seen too many people put in electronic batch records, for instance, that tie to individual unit operations, and then they pile on of, oh, I want this data available for my supply chain so they know right where the manufacturing process is, and we want to get data, and all of a sudden your IT network is overwhelmed and it's taking 6 hours to run a step that should take 30 minutes. I might exaggerate slightly, but really spelling out what do you want to build to over the next few years so that you get the infrastructure right. And I think the second piece is exactly that. When you're building into the process, what parts of your process are likely not to change? Focus on those. What are the key areas? They may change amounts, but media prep, pluripotent stem cell expansion, those things are typical. And once you get it right, you should be able to automate. And what steps do you just not understand yet? And don't spend the time there. Build a block so that you can really learn the process understanding and then try to automate. I think those are the keys, is ask what you're trying to get out of the automation, because it's so much cheaper to do it right the first time than reworking that work while you're launching a product.
Absolutely, for sure. To from a cost perspective, especially to get it right the first time. Narinder, I want to give you the opportunity to provide some information here too. Any additional thoughts on best practices or things to avoid from a process development standpoint?
I think Craig hit the nail on the head. It's all about automation, not just for automation's sake, but really understanding what are you trying to do here, having a deep understanding of what drives the cost in this case, or inefficiencies for your process. For example, we saw that in our case, reducing process cycle time by 20% for one of our processes had a greater than 30% impact on plant capacity. Well, so understanding what drives plant capacity and focusing on that specific thing. So reducing it further than that was actually not as much benefit. So it's not just absolutely going down to what needs to be, what its minimum could be, but really finding the sweet spot, right? There's a cadence to execution. Getting it right first time. This is still a very custom manual processing here, and really understanding sources of variabilities. We are collecting material from very sick patients in various different stages of diseases, and that variability is tremendous sometimes, especially in the real world once you go out of clinical trials. So how can we define a process where we understand these sources of variabilities from the patient material, from the reagents, from the process itself, and then building a robust process around it? And then getting the automation right, like Craig mentioned. Can you think about not just the process, but analytics as well? Think about electronic batch records. Also integrating QC and QA in a way that can reduce mistakes. The other areas we've been focused on is finding automation to reduce any manual open processing steps so that you can eliminate any contamination risks. And also think about how can you utilize more efficiently the expensive reagents and vector products. So those are the things we are focused on.
If I could just interject one other point, you also
Sure.
mentioned earlier when you're using CDMOs, they're an expansion of you, but it's also key to understanding what their expertise or what their weak points are so that you automate for the hands that are going to be using the process.